Hi, good morning, everyone. Welcome to join our chemistry webinar by today. So, Mr. Akara, and I think we can start it now.
I think we're having a network issue there. Welcome everyone. Good morning, good afternoon, good evening, wherever you are across the globe, especially the West and East Africa, to this very special webinar. Maybe when Dr. Jits Okara is back, the Registrar and Secretary General of West Africa Postgraduate College of Medical Laboratory Science, Abuja, will give us his opening speech. Once again, we want to welcome every one of us that make it a day today. My name is Dr. Abiodun Olaya Paul. I'm the co-host, and I'll be supporting Dr. Kara to moderate this webinar. So once again, we want to welcome you. I don't know if Dr. Kara is there. I can see him talking. Dr. Kara, are you back, sir?
Yes, I am.
Over to you, sir.
I am very sorry. The network has been very challenging here. Be that as it may, it's my privilege and honor to introduce Dr. Paul Olaya, who will be standing in for me because of another emergency commitment that I have in Abuja. So please, you will permit me to hand over to him to play the role of co-host. I have no doubt in my mind that it's going to be a very fascinating and interesting program. Thank you very much indeed, and have a nice webinar. So over to you, Dr. Paul Olaya.
Thank you, sir, Dr. Gisi Akara, for that opening speech. So once again, we want to welcome every one of us to this special, I call it very special webinar. We'll quickly look at the agenda of what we're doing today. Of course, Dr. Gisi Akara have given the welcoming opening speech, and we'll be following that with the first speaker by Dr. Yetunde Sorian, which will be talking about the early diagnosis of renal glomerular dysfunction, quality issues, interpretation of plasma creatinine results. And that will be followed by Dr. Tim Yin, who will be speaking on biochemical analysis, better medical practice to COVID-19 treatment. And we'll be wrapping up the speaker with Ms. Akeleshi Ekwanwa, very special person, will be taking us on Mydrake chemistry solution introduction. So that will definitely follow with question and answer. So as it is now, you permit me to welcome our first speaker, Dr. Oyetunji Osorian, which will be speaking on early diagnosis of renal glomerular dysfunction, quality issues, interpretation of plasma creatinine results. Dr. Oyetunji is a senior lecturer and a very strong consultant chemical pathologist at Lagos State University Teaching Hospital. He's also a consultant chemical pathologist at SYNLAB Nigeria. So join me, welcome Dr. Oyetunji Osorian for his presentation. So thank you so much.
Good morning, everybody. It's a pleasure to be given the privilege to give a talk at this wonderful occasion, this webinar. And I am Oyetunji Osorian, consultant chemical pathologist and the head of department of clinical pathology in the Lagos University Teaching Hospital. I also am a senior lecturer at the College of Medicine, University of Lagos, and I also consult on part-time basis for SYNLAB Nigeria. So it's my pleasure to talk to you this morning on the topic of early diagnosis of renal glomerular dysfunction, quality issues in the interpretation of plasma creatinine results. So the outline as a brief introduction and snapshot of chronic kidney disease in Nigeria, and a brief overview of the biochemical test of renal function that will go through the glomerular test, renal function test proper, and the estimated glomerular filtration equations, and then we'll talk about why early diagnosis for renal glomerular dysfunction. Why is this important for us as laboratorians, as clinicians, to be aware of this? And then zoom in on quality issues with plasma creatinine assay and the estimated glomerular filtration rate estimation and interpretation. Vis-a-vis the issue about need for standardization and traceability. And what are the way forward? Then we'll summarize and move on. So by way of introduction, renal glomerular dysfunction is usually asymptomatic until quite late. By the time someone is manifesting clinical signs and symptoms of renal glomerular dysfunction, it's actually late in the day. So most of the symptoms and clinical manifestation can be non-specific also, like fever, for example, bloody urine can be due to a bladder infection, a lower UTI infection, and or a kidney stop. So it can be non-specific, weakness, malaise, feeling tired, low blood level, or anemia. All this can manifest in several other diseases. So that's why high index of suspicion is needed, and for early diagnosis to save time, because late diagnosis in the context of renal glomerular dysfunction, actually chronic kidney disease, can be very expensive consequence to manage, both for the individual and the national health budget. Chronic kidney disease require renal replacement therapy, continuous hemodialysis, and then the definitive treatment is kidney transplant, which is very expensive, and even after doing the transplant, you need to sustain the graft using immunosuppressant, and that can be cost capital intensive also. So early diagnosis is key. We'll talk about urinalysis, glomerular and tubular function tests. These are probably the categories of renal function tests available. So those are the three categories of renal function tests: urinalysis, glomerular and tubular function tests. Another thing to note is that in end-stage renal disease, that end-stage phase is very challenging and difficult to determine the initial cause. So it's better you diagnose, you pin down the etiology, the cause of that renal problem before the patient enters that end-stage renal disease phase, which is the last phase in the spectrum of acute kidney injury and chronic kidney disease. The last phase in that spectrum is end-stage renal disease, and of course, it's irreversible and will require a transplant or some form of renal replacement therapy in terms of dialysis, either hemodialysis or continuous peritoneal dialysis, pending when a transplant will be available. So you don't want a patient to get to that stage. If you have seen a patient at that phase, it then becomes very difficult to know what is causing this. And that's important because if you do a renal transplant, for example, if the etiology of that renal disease is due to an immunologic problem, maybe immune complexes, autoantibodies, the same immune complexes, autoantibody that damage and kill the first kidney will ultimately damage and kill the newly transplanted kidney also. So it's important to pick them up early so that you can make the diagnosis early and the etiology, and then address that. If the patient comes in, unfortunately in our environment, research data is showing that most of our patients present quite late here. That's why the more reason why we need to sensitize ourselves on the need to diagnose these patients early. So chronic kidney disease is a long-term condition resulting from inherited renal functions, commoner in the elderly. Physiologically, normally, the kidney function deteriorates gradually with age as you're aging. So that's why it's not surprising it's commoner in the elderly. And of course, it's more common for genetic reason and reason not poorly understood yet, more common in Blacks and the Southeast Asian populations. So a quick snapshot of the burden of the disease in Nigeria. Prevalence from data available varies depending on where the study was done, and the environmental prevalent condition varies between 11.4 to 26% in different studies that have been done. And then in a systematic review that was done in 2018, meta-analysis for the whole sub-Saharan African continent as a whole put the prevalence as 13.9%. So various studies were with different results, possibly because, as some of the studies highlighted, the patient with mild to moderate cases of chronic kidney disease are grossly underreported or underestimated in our community here. And that's why we need to sensitize ourselves to be able to diagnose these conditions early before they enter the irreversible phase. In our setting and worldwide, the commonest cause of chronic kidney disease is hypertension and diabetes mellitus. But in terms of our own local profile of causes of chronic kidney disease, as highlighted, hypertension, top the list, diabetic mellitus, and here in Nigeria, about 37% of chronic kidney disease patients actually will have diabetes, have been found to have diabetes mellitus. HIV also has added to the burden. HIV nephropathy has added to the burden, and there was a study that was done in 2010 that pretty much made the case for early diagnosis of chronic kidney disease. From this particular study, chronic kidney disease accounts for about 8 to 10% of all of hospital admissions. And these patients have, most of them have advanced, they present late, so they are not picked up early. That's a huge challenge for us in our environment here. So the prevalence of mild to moderate CKD in the population has not been well established because of this underreporting issue. So renal replacement therapy, of course, you know, is expensive in our environment. For a confirmed renal failure case, we need at least minimum of 3 sessions of dialysis per week. That costs between 20,000 to 30,000 per session, and you need minimum of 3, and sometimes it can be more than 3 in a week, and that is for life, at least till a renal transplant is done. So it is better avoided. It's a big financial burden. Even when you get transplant done, transplant is quite expensive on its own. Immunosuppressants are quite expensive to sustain the newly transplanted kidney after the surgery. So it's better avoided, and that's why we need to diagnose this condition early, and that is the main objective of this webinar, to sensitize everyone, all the stakeholders, the laboratorians, the physicians, and the general public on the need for early diagnosis. So early diagnosis require educating the public, the patients, the clinicians that see these patients, and then accurate clinical chemistry testing, accurate measurements. Is key for making early diagnosis. So I'm going to quickly go to this case just to, as an introductory case, a 45-year-old business executive that was seen in the emergency room with a 5-day history of vomiting repeatedly, facial puffiness, bilateral pida, swelling, 3-day history of anuria, 2-day history of restlessness, agitation, psychotic behavior, vis-a-vis hallucination, visual hallucination. The patient happened to be a known diabetic and hypertensive, and plasma urine electrolyte result done by the general physician, the family physician monitoring this patient has all been normal over the previous 1 year. And at this particular presentation, the electrolyte urine creatinine result has shown hyponatremia, moderate hyperkalemia, metabolic acidosis, azotemia, and a markedly elevated level of creatinine. The urine creatinine is suggestive of some form of renal glomerular dysfunction. So, why was this? The question is, why was this renal glomerular dysfunction not diagnosed earlier? Please, can you co-host mute those who? Can you mute those? Okay, thank you. So, the question is, why is renal glomerular dysfunction not diagnosed earlier? This is a known, the 2 commonest cause of chronic kidney disease, is happened to be diabetes and hypertension, and this is a patient with diabetes and hypertension. But over the past 1 year, at least from records available, it's only urea creatinine and electrolyte that the physician, the family physician, was doing to monitor his progress as far his renal status. And this is one of the points we need to make in this webinar. This patient now came down with uremia. Uremia is the end of the spectrum. The patient has decompensated fully and is a full-blown chronic kidney failure. So I bet this patient shouldn't have ended up like this, because at this phase it becomes irreversible. The patient has to be on continuous dialysis, renal replacement therapy, pending when a transplant will be available, and that can be even to get suitable donors, matched donors. They go through some HLA testing and cytotoxic T-cell cross-match testing to match. You need a matched donor. Those tests are very expensive. HLA, cytotoxic T-cell tests, you spend nothing less than between 200 to 300,000 naira. That's a lot. That's over $500 just for one test, and that test might still come out to be not matched. You need to still do other tests till you find a suitable match. So even the price of getting a donor, a suitable donor, is very expensive. Not on top of the surgery itself, the cost of immunosuppression. That's what this patient is going to be burdened with now, but it's very avoidable if the physician has just done the right test. For a patient with risk factors of chronic kidney disease like hypertension, diabetes, they're supposed to be monitoring at least a test that assesses the glomerular filtration rate once every 3 months. Glomerular filtration rate will have picked up this renal glomerular dysfunction before this patient decompensated. Before urea creatinine, this is a common practice in the population. Before urea creatinine is elevated, the patient must have lost about 50% of the renal function. So already the whole kidney is gone by the time you start seeing elevation of urea creatinine. So it's a bad practice waiting for urea creatinine to be elevated before you are alerted to the existence of some ongoing renal glomerular dysfunction. So this physician was rather focusing on urea creatinine, to be waiting for it to be elevated before he or she will be alerted to the fact that this patient's renal function is deteriorating. And that's why this patient ended up like this. If you had done maybe a creatinine clearance or estimated GFR, well, months ahead, they will have picked up this dysfunction well ahead of time. You will see a graph I'll project to buttress that point soon. So just a quick overview of the biochemical test of renal function. Why do we need to do that? To identify renal dysfunction, diagnose renal disease, monitor disease progress and response to treatment, and of course to assess changes in renal function that might impact on therapy. For example, some drugs like digoxin and some cancer chemotherapeutic drugs, they are mainly excreted from the body through the route of the kidney. So if the patient ab initio, before starting such treatment, has a compromised or suboptimal kidney function, and you're going to give such a patient such drugs that's going to be majorly eliminated through that route, then you are going to have serious issues, because a normal therapeutic dose, a safe dose, will then become toxic to such a patient because of increased half-life in the blood, because the kidney that's supposed to eliminate that drug is not working well. So a tiny amount of the drug that's supposed to be very safe for the patient will then become very toxic. That's one of the reasons why you need to determine the renal function prior to initiating such therapy. So about signs and symptoms, I won't dwell much on this. Like the patient we saw had uremia, nausea, vomiting, some multi-organ dysfunction like uremic encephalopathy, responding for the psychotic behavior of that patient. The patient had anuria, lack of urine production. So problem of urine volume. Some patients can present with polyuria rather than oliguria or anuria, which are reduced urine volume. They might even have polyuria. So it varies, and depending on the cause, etiology, some might have some protein in urine, some blood in urine, and then edema, pedal facial, like that patient had facial puffiness and pedal swelling. That's edema because of low protein level. So the biochemical test of renal function, like I said earlier, the introductory slide, that broadly divided into 3: urine analysis, also called urinalysis, and then measurement of glomerular filtration rate, and then tubular function test. The commonest, the most commonly done amongst all these are the test of glomerular filtration rate, measurement of glomerular filtration rate, that the commonest test of renal function. So glomerular filtration rate is essential to renal function. It's the most frequently performed test, and it's based on the concept of clearance, which is the determination of the volume of plasma from which a substance is removed by glomerular filtration during its passage through the kidney. And that's the formula to calculate the clearance of any substance. The most commonly done clearance test in terms of estimation of GFR in the routine hospital setting is the creatinine clearance, and which we are going to talk about in more details soon. Listed in the last bullet are the properties of an ideal marker of glomerular filtration. You want a substance that is freely filtered and only eliminated from the body through the kidney, a non-toxic, easily measurable, a substance that is not protein bound and is not reabsorbed or secreted from the renal tubules, the nephrons. So these are some of the commonly used markers of GFR. The first two are the most commonly done in a routine hospital setting, while the last two columns are the gold standard. These are not practicable for routine, everyday hospital laboratory purposes. The inulin clearance and the isotopic clearance, these are the gold standard, and they are the tests that are used to standardize the creatinine and other analyte like urea that we use in a normal hospital setting. So those are the properties of an ID marker as highlighted. You can see that we don't have an ideal marker. Though we have gold standard, we don't have an ID marker. Because even though inulin and the isotopic ones are very good, they are the gold standard, they are still not the ID, because one of the properties of an ID marker is that it should be endogenously produced, and those two substances are foreign to the human body. They are not endogenously produced. I won't dwell much on this. I've mentioned that this is the gold standard inulin clearance, is the gold standard with the isotopic one, and of course it is the continuous version, the continuous infusion inulin clearance that we're talking about in this concept. Because there's another type of inulin clearance called the bolus, that's as a bolus. That is not the gold standard, that is a silver standard. Isotopic agents can be used as highlighted: chromium 51, EDTA, and technetium 99, diethylene, triamine, pentaracetic acid. These are, however, not practicable for routine hospital laboratory use. They are only meant for standardization and research purposes. So creatinine clearance, of course, is the commonest test of glomerular function done in terms of assessment of GFR in the routine hospital setting. Usually timed collection over 24 hours, and then a blood sample must be taken during that period of collection, because for you to calculate the clearance, you need some variables, the U, V, P that I showed earlier, the equation I showed earlier. That P is the plasma concentration of that analyte. In this context, creatinine concentration in the plasma, and then the urine concentration is the U, and then that V is the flow rate of urine. So you need a corresponding blood sample to be collected during that 24 hours. However, this process has its challenges. Practical problem of accuracy of urine collection. Some patients might miss during that 24-hour period. Maybe they are in the bathroom or in the restroom doing a major, and there is what we call reflex micturition that do take place, and then they forget to bring the container with them into the bathroom or the restroom, and then so they miss collecting some volume that should have been accounted for. So that can cause some inaccuracy and variability. Of course, creatinine as an analyte itself have a high within subject variability for different reasons, and of course the problem of analytical interferences in the measurement process, or the method for determining the creatinine level. Creatinine clearance overestimate GFR compared to in the gold standard inulin clearance, because it is secreted by the tubules, though this can be inhibited by giving the patient cimetidine, but it's not practicable to give every patient cimetidine. And then it overestimates GFR more in frank renal failure, because the higher the level in the blood, the more it's secreted into the urine, and that's the confound your calculation of the clearance. So it's time consuming, of course, inconvenient, and issues about accuracy of the result based on several factors that can interfere in the assay. We know that creatinine is a product of muscle metabolism. It's an iodide of the muscle protein creatine, which is converted to creatinine either through direct hydrolysis or through the route of creatinine kinase. Either way, you end up with creatinine. So it's freely filtered at the glomerular, so it's a good representation of the glomerular filtration rate. And unfortunately, because it's a product of muscle metabolism, muscle mass has a bearing on the level. So its baseline is higher in males because males are naturally more muscular than females. And I've mentioned that some tubular excretion takes place. So interpreting plasma level of creatinine can be due to several factors. This is just to help us understand that this is a normal situation because the level of creatinine in the blood is a balance between production rate of production and the rate of elimination through the kidneys. A rate of production in the muscle, that's a normal, so this is a reference interval, normal range of creatinine. However, creatinine can be elevated in the blood when you have diseased kidneys because of a reduced rate of elimination. It's normally filtered and eliminated through the kidney. So when the kidney is diseased and renal glomerular dysfunction, it accumulates because of the diseased kidney, even though there is no change in the rate of production, normal muscle mass. It can also be elevated even with a normal kidney function. There's nothing wrong with the kidney, but when there's increased muscle mass, like in the bodybuilders, sports men physiologically have more muscle mass, so they have baseline higher creatinine, and in some muscular dystrophies, some disorders that cause rhabdomyolysis, breakdown of muscle, and some genetic disorder like Duchenne's muscular dystrophy, all this can cause, or intramuscular injection can cause elevation of creatinine in the blood, even with a normal kidney function. So when you see creatinine being high, it doesn't always translate to a renal glomerular disease. You have to look at other factors like the muscle, the metabolism. And when you have a problem about the kidney and the muscle, of course, you have elevated creatinine in the blood. So quality issues with plasma creatinine assays. The accurate estimation of the glomerular filtration rate using formulas that have been validated with and correlated with the gold standard, the inulin and isotopic clearance are now available to bypass some of these challenges of accuracy of collection, the inconvenience of peripheral urine collection. The patient has to be grounded the whole day to collect that. So, you know, investigation of chronic kidney disease is highly dependent on the measurement in the clinical chemistry laboratory. For you to use any of the estimated glomerular filtration rates, which for most of these formulas, you just need to have a plasma creatinine result, and then you put in some parameters like the age, the sex, depending on which formula you are using, and then to estimate the GFR, and it's pretty much accurate. As long as the patient is in steady state, as not decompensated, is a true representation of the GFR. We'll see a few of those soon. However, for you to use those formulas, you need to have measured creatinine accurately. And the guideline is that you should, in fact, most of those formulas were derived from studies that use creatinine assay method that have been highly standardized and that are traceable to the reference method, which is the isotope dilution mass spectrometry method. That's the reference method for measuring creatinine. So any method that you want to use to generate a creatinine result that you are going to use to estimate using those equations should be traceable to that reference method. So in terms of the guidelines to that make a diagnosis of chronic kidney disease, you need a GFR, glomerular filtration rate estimation, either through direct creatinine clearance measurement or estimated glomerular filtration rate using the plasma creatinine result, and then in addition, urine albumin to assess damage to the glomerulus causing leakage. So these are the two key analytes we need to investigate chronic kidney disease. And these are the various stages of chronic kidney disease. You can see that the GFR is what is used to classify the various stages. So if you do not have accurate measurement of GFR, you can misclassify and mismanage the patients. In terms of albumin, these are the KDIGO, the kidney disease improving global outcome guidelines, and that guideline is actually like the Bible of chronic kidney disease and acute kidney injury. So why do we need accurate laboratory methods for estimation of creatinine? For the same patients, I may have measuring the creatinine in different lab using the same sample. The result might be varied because of lack of standardization. So the goal is to make sure that there's comparable results, irrespective of the platform that we use to analyze that creatinine, once the guidelines are followed in terms of ensuring that the method you are using is standardized, is traceable to the IDMS, which is the reference method. So classification of chronic kidney disease depends on specific cutoffs for EGR, as you saw in that list. The various stages I listed, it depends on the glomerular filtration rate and the urine albumin. So these cutoffs have been derived from serum creatinine, urine albumin methods of the highest quality. So inaccuracy in the measurement of creatinine and albumin will lead to misclassification and mismanagement of such patients that can compromise patient safety. That you want to avoid. So although serum creatinine and urine albumin methods are simple to perform and cheap, they are very quality because of issue of standardization. So these are the various methods available, just a snapshot of methods available to measure creatinine, which is our focus for today. The most commonly used method is the Jaffe method, which has been around for over a century. It's very simple, inexpensive to perform on most platforms. However, it lacks specificity and can give high results compared to the reference method, which is the isotope dilution mass spectrometry. And then this interference make it difficult to use in children where normally they have low muscle mass. The creatinine levels are baseline lower than that of adults because of their muscle mass. And then in pregnant women and also. So for this reason, compensated methods that have inbuilt adjustment to the calibration to minimize interference have been developed. So we have compensated Jaffe method. So whatever method you are using in your lab, make sure it's traceable to the reference method IDMS. And if it's a Jaffe method, that better still, it is the compensated Jaffe method that you are using. That has minimal interference compared to the routine Jaffe method. Enzymatic methods also exist using creatininase and creatine amino hydrolase. Some of the enzymes that can be used, they are more specific than the Jaffe method, and results are traceable to IDMS, and they are available on many commercial platforms. However, they are more expensive compared to the Jaffe method. Of course, chromatographic method also exists, and the reference method is the isotope dilution mass spectrometry after chromatographic purification. And this has been calibrated with a primary reference pure creatinine materials, which can be obtained from the United States National Institute of Standards and Technology. Commercial methods that we use in the labs have been calibrated in line. I expected, let me put that way, to have been calibrated in line with the primary reference material. And these are the ones that are said to be traceable to the IDMS. So what the KDIGO guideline for chronic kidney disease advocates is for use of estimated glomerular filtration rate, which is desirable now to bypass all the issues with collection of 24-hour samples and the inaccuracy and all that comes with that. To bypass all that, just do an estimated, advocated for estimated glomerular filtration rate to be done. And for you to do that, you have to make sure that the method you're using to generate that creatinine result you're going to use in that equation to estimate the GFR is traceable to IDMS. So the use of non-traceable methods invalidates the derivation of estimated glomerular filtration rate. So it's not just, and that's one of the commonest causes of, the lack of consistency in results. The same patient goes to one lab, gets a result, goes to another lab and gets a different. The clinician, the nephrologist is confused. What is happening? He wants to know what is the correct state of the renal tissue of this patient. So the lab has that onus to give them reliable tests, and that can come when we make sure that the method we are using is traceable to IDMS. And if it is Jaffe method, is the compensated Jaffe. And if the enzymatic method, whichever one, they must be traceable to IDMS for you to have a valid estimation of the glomerular filtration rate. of course, we know that the reference method is impracticable for routine hospital use. It's meant to calibrate the routine hospital method. In this UK NEPWAS study, you can see the true creatinine value of that sample, 81. And it was sent to several labs that use different methods and different platforms to analyze the creatinine of in that sample, and look at the result varied from 70 down to 118, but the true value is 81. So, and that was then in 2006, very little standardization existed at that time. The awareness was very poor generally, so most labs just use whatever comes without ensuring that it's traceable to the reference method. However, by 10 years later, by 2016, when the same survey was done using the same, another sample containing the same amount of creatinine, 81, you can see that you have better correlation now, because at that time, most labs, many more labs are trying using methods that are traceable to IDMS, like the enzymatic method. As at 2006, there's only one enzymatic method user available in that particular survey in the UK at that time. Then by 2016, several over 100 labs are already using those methods, and that is what accounted for a total. I can see the variation is less compared to the previous one, 70 to 118. The variation is minimal here, and the mean of that all the method was 80, which is close to 81, which is the true value, compared to the mean of 93.6. Those methods that were not traceable that were not well standardized, that were used previously. So that just buttresses the need for standardization of the creatinine method that we use. So we've talked about this. I made this point earlier. In that case that we had, the introductory case, reference ranges can be misleading. We should not wait for creatinine, plasma creatinine urea to be high before you are alerted to the extent of renal glomerular dysfunction, because the creatinine in the blood will not be elevated. You have lost minimum of 50% of your kidney function, your nephrons, functional nephrons. However, if you have done a GFR assessment, GFR measures precisely the amount of functional nephrons, so irrespective of plasma creatinine level. So a GFR assessment will have picked up the dysfunction much earlier for that patient that we saw. But the doctor in that case was waiting for urea electrolyte and creatinine to be high before he or she is alerted to the existence of renal glomerular dysfunction. So note that reference ranges can be misleading. You should not wait for creatinine to be high. And this is what this slide here is projecting. Waiting for plasma creatinine to be high to diagnose chronic kidney disease is misleading. If you look at this graph, the upper limit of the reference, or the upper limit of normal, is the one up here. The lower line is the lower limit of normal for creatinine. That's a reference range or reference level for creatinine. And you can see on the x-axis here is the glomerular filtration rate, the creatinine clearance estimation. So you can see that from this first point here, 120, close to 120, the patient has lost close to half of the kidney function. However, creatinine is not yet elevated, it's still within all through that period. Over those several months, it's still within the reference interval. It's not yet elevated. So if the doctor, like in our case, over that previous 1 year, has been doing electrolyte urea creatinine, they've all been normal, and he went to sleep thinking there's nothing wrong with the kidney of that patient, not knowing that the patient's renal function has deteriorated significantly until the patient came down with frank uremia, which is the end phase of that spectrum. So that's what happens when you wait for creatinine to be elevated before you know a patient has a kidney problem. So the best way for early diagnosis, the best test for early diagnosis of renal glomerular dysfunction is an assessment of glomerular filtration rate, either to the creatinine clearance, or better still, as advocated currently, the estimation of the glomerular filtration rate using a plasma creatinine result that you now convert to a glomerular filtration rate in mils per minute, using any of those equations that are available for estimating GFR using plasma creatinine. Because waiting for creatinine to be high is misleading. We lose a lot of valuable time, and then the patient risks entering the irreversible phase, which is very expensive for everybody. However, by the time creatinine becomes elevated in the blood, it becomes a sensitive index of any small change is accompanied with a similar change in GFR, so it becomes an accurate representation. So by the time the patient is fully decompensated, like that, our patient, the creatinine of 450 micromoles per liter, it doesn't need any estimate of GFR anymore. If I estimate of GFR with a decompensation decompensated patient is misleading because the equation was validated for steady state. By the time patient is fully decompensated, those equations, they lose their sensitivity. They are not meant to be used in a compensated patient. They are meant to be used in steady state patient. Those are the formulas we are talking about. These are some of the analyses that can interfere in estimation. And years ago, one of our professors in pediatrics in my teaching hospital called the lab, and I was the consultant on call that day about a creatinine result of one of his patients that had recovered from acute kidney injury. The patient child was doing well, and then the result was quite high. So he said it doesn't correlate with clinical picture. So he called me, and I asked him, what drugs are this patient on? Because I knew the commonest in our environment, cephalosporin, for such condition that the patient had sepsis, third generation cephalosporins are commonly used. And cephalosporin antibiotics can cause positive bias or positive interference or cause overestimation of creatinine. And that was the reason that in the form they sent to the lab, they did indicate the drug the patient was on. And that's why filling that form for doctors is very important to fill the form accurately, because it will help the lab to give you the best result. If they have indicated on the form that this patient is on cephalosporin, we will look for an alternative method that is not prone to this interference. We would have used the Jaffe method, which is the commonly used method worldwide in most labs, and that is the method that is prone to all this interference as listed. Then negative interference at bilirubin and hemoglobin, they can cause underestimation of creatinine. So creatinine clearance of course is inaccurate because of incomplete bladder empty. Some patients might have prostate issue, enlargement of the prostate that cause incomplete voiding, and that volume that's supposed to be in the container, some are still retained in the bladder. So that will add to the inaccuracy. So wide individual variation, burdensome to the patient. That is what led to the use of the formula. The formulas that have been validated and that are recommended now by the clinical guidelines is the chronic kidney disease epidemiologic equation, CKD-EPI, and that's the most validated now. Before that is the MDRD, the modified diet in renal disease patient formula, that was the most validated in that population of chronic kidney disease patients. However, the CKD-EPI formula equation, all these are available. You don't need to memorize the equation. That's why I think about giving you the details of the equation, because you can download the calculators. Online calculators exist, Android apps exist. Just put in the creatinine Victor, they will estimate the GFR for you. Once you give the age of the patient, the weight, and all that. So depending on which of them, but the most validated and the best of all these formulas are the chronic kidney disease epidemiologic equation, CKD-EPI. And so problems using plasma creatinine and the formula, like I said, the formulas applicable only in steady state. When the patient decompensates, they lose sensitivity. We have mentioned a lot about issues with plasma creatinine and interference in that. Patient with muscle wasting, drugs that inhibit tubular secretion can raise creatinine concentration, edematous patients and all this. So the alternative to creatinine now exists in terms of cystatin C, the cysteine proteinase inhibitor C, a low molecular weight analyte produced from all nucleated cells in the human body, freely filtered at the glomerulus, is more accurate as an estimation or as an estimate of the glomerular filtration rate. It better reflects the glomerular filtration rate than creatinine. However, it's more expensive, and it's not influenced by muscle mass, diet, sex, like creatinine. However, the drawback why it's still not the ideal marker is because it's mostly reabsorbed in the proximal tubule. But in the end, this reabsorption doesn't really affect the usefulness, because most of the cystatin that's reabsorbed into the tubular cells are completely metabolized inside the cells. So they don't enter the blood. They don't get back to the blood as cystatin C. So that's why it's still pretty much the best marker we have now, endogenous marker of renal function, renal glomerular function. So because of this cystatin C-based chronic kidney disease EPI equation and MDRD estimated GFR formulas now exist. However, like we said, drawback to this is that it's very expensive, and it also has its own issues in people that are smokers, and hypothyroid thyroid dysfunction affect the result of cystatin C. I was privileged to be one about the very first in at least this part of the world in terms of Sub-Saharan Africa to measure cystatin C, and that paper is available in the journal, International Journal of AIDS, HIV/AIDS, and that was over 10 years ago. Okay, so lastly, tubular function. We've done glomerular function test. The last renal function test, tubular function test. Those are not our focus for today, just for completeness sake that we are listing all these proximal tubular function tests and tubular function tests. Why early diagnosis made all this point? Mild to moderate chronic kidney disease largely undetectable in underreported in Nigeria. Patient present late in Nigeria with severe end-stage chronic kidney disease commonly, and that is associated with a high death rate. And then the financial burden of chronic kidney disease is very huge. Renal replacement therapy is not cheap. Hemodialysis costs close to about 25,000 to 30,000, depending on where it's done per session. And a patient needs minimum of 3. It can be more, but minimum of 3 every week, and that's for life, or at least till a kidney transplant is available. And the transplant surgery is very expensive. Immunosuppressants are very expensive, which you need to sustain the newly transplanted kidney after the surgery. So it's a very expensive process. It's better avoided. That's why we need to make early diagnosis so that people don't end up in that irreversible phase of end-stage kidney disease. So, and also the progression from mild to moderate to severe chronic kidney disease can be reduced by simple, inexpensive lifestyle changes. For example, low protein diet, monitoring their blood pressure, their glucose level, and then use of, for those that have mild proteinuria, you can put them on ACE inhibitors or angiotensin receptor blockers. They've been shown to reduce progression to chronic phase or even abort it altogether. So, and these are very cheap to do. So if you can pick it up early, we can intervene and slow or abort progression to that irreversible, chronic, very expensive phase. That's why we need to diagnose early. The last case I'll give is a 56-year-old woman being managed for chronic kidney disease who had a renal transplant done 9 months earlier in the United Kingdom, was seen in the renal clinic on follow-up visit with a plasma creatinine of 200 micromoles per liter that was done while she was in Nigeria during the COVID-19 lockdown. And at that time, she couldn't travel for a follow-up visit in the UK to see a nephrologist. So at the last plasma creatinine result that was done 6 months earlier in the UK was okay, 105, which is okay. The nephrologist then requested for a repeat based on this markedly elevated 200 from 105 increase to 200 within a 3-month period. So the nephrologist asked for a second opinion, and this was done in the UK. The result came as 105, which correlated with the one that was done 6 months earlier in the UK, the last time she came to the UK before the COVID pandemic hit. That was 105. The result of the repeat was 100, which correlates. So what is the explanation for this discrepancy? Why this 200 all of a sudden? One of the problem is that many laboratory measurement procedure for creatinine give different results for the same specimen because of lack of standardization, because several labs are using methods that are not traceable to the reference method, which is the IDMS that we talked about earlier. So you have discrepant results of plasma creatinine for the same patient. And a patient with transplant coming with a high figure like 200 that the patient came with, that connotes a very serious issue, because the physician and nephrologist want to immediately know, is this rejection that is taking place? Is this new kidney being rejected? Is it renal transplant rejection that is taking place? Or is this effect of some of the immunosuppressants that we give those transplant patients? They are nephrotoxic, like cyclosporine, for example, is nephrotoxic on its own. That's why any patient on such drug should have therapeutic drug monitoring being done. They monitor serially the level of the immunosuppressant in the blood so that it doesn't, they make sure it's within the therapeutic range, because it has a narrow therapeutic range. So the physician need to know what is happening. Is this rejection of the kidney that's a newly transplanted kidney, or is this side effect of the immunosuppressant? Do we need to reduce the dose of the immunosuppressant? Are we giving too much to the patient? And that poses a lot of challenge. However, so they get confused. That's why you ask for a repeat, and the repeat done correlated with the previous sample that was done in the UK. What was the issue? The lab that generated that result in when the patient couldn't travel to UK for a follow-up used a method that that was not standardized. So the results will be, because it's not traceable to the reference method, the results will be different. So standardization is just achieving comparable results by having calibration traceable to a reference measurement procedure. In this case, the reference measurement procedure for creatinine is the isotope dilation mass spectrometry, IDMS. So every method used, Jaffe or compensated Jaffe method you're going to use to estimate creatinine should be traceable. So check the product information leaflet that you have for that reagent you're going to use to note whether it is traceable to IDMS or not. If it's not traceable, reject it. Buy from suppliers that supply standardized kits that are traceable, so that you don't have this kind of challenge that the patient is having discrepant results, and that can be costly in terms of clinical management of the patient. So it must be traceable. Traceability refers to the ability to link the calibration of the laboratory test back to the reference measurement procedure using a continuous chain of reference. What is the way forward? Standardization. We need to standardize the laboratory creatinine assay methods and practices. Of course, clinical collaboration also is still needed. The clinicians should not wait for elevation of plasma creatinine from the electrolyte urea creatinine result before being alerted to the existence of a chronic kidney disease or before diagnosis of renal glomerular dysfunction. The clinician should do an assessment of GFR. For every patient that's hypertensive or diabetic, the recommendation is you have a GFR test done minimum at least once every 3 months. So that will pick it up earlier than waiting for the urea creatinine to be high, which will not happen until the patient has lost 50% of the kidney function. So that can be devastating. So clinicians should request for estimated GFR estimation from a laboratory that uses standardized plasma creatinine method that is traceable to the isotope dilation mass spectrometry reference method. Okay, I won't waste time here. This is just to elaborate further on the two themes going forward: clinical collaboration with the clinician, and then standardization, that's in the purview of the lab, standardization of the method and practices at the national level, the state level, and the local level. So in terms of the national level, as I highlighted, these are the necessary steps. Collaboration is needed between the Association of Clinical Chemists of Nigeria and other associations that are stakeholders in the lab, the Pathologist Association, the Medical Lab Science Association. Should collaborate with the Nigerian Medical Association, the Nigerian Association of Nephrologists in the context of chronic kidney disease that we're talking about here, to create awareness, the need for clinicians not to wait for elevation of plasma creatinine before saying the patient has a kidney issue, and to do estimated GFR for the patient early enough, and then to assure them that they are going to use standardized methods that will make the estimation of GFR that they're going to be using in the clinic effective and accurate and clinically relevant based on available guidelines. In terms of the lab, the leaders of the clinical chemistry and laboratory medicine should collaborate with leaders of for nephrology, and areas of collaboration include agreements on the formula to use, the standards for to standardize the creatinine method, and training of and sensitization of laboratory personnel and renal physician on the need for early diagnosis of renal glomerular dysfunction, and at the local level, need for reporting serum creatinine and estimated GFR routinely. Any person that has a serum creatinine in the lab, you should calculate as a lab the estimated GFR and put it in the result you are sending out, the report you are sending out, so that it will be more clinically relevant and useful to the patient and the physician managing the patient. So in summary, renal glomerular function tests are the commonest renal function tests. Clearance tests are better than plasma creatinine urea for early detection of renal glomerular dysfunction. There is a need for standardization of laboratory methods and practices for creatinine assay to ensure that they are traceable. Clinicians should request for estimated GFR or creatinine result from a laboratory that they have confirmed is using a method that is standardized, that is traceable to the reference method for estimation of plasma creatinine. Further information you can get from the KIDIGO publication. I want to acknowledge Dr. Graham Biscoe, the past president, International Federation of Clinical Chemistry and Laboratory Medicine, who provided some of the data I used. Thank you all for listening.
Yeah, thank you so much, Doctor, for that wonderful presentation. I think you've really done justice, Dr. Yetule Soriya. That was very, very beautiful. I think we'll try as much as possible. Please, if you have questions, try as much as possible to put your question in the box, so that we can all see what you look at the questions, and I'm sure the speakers will also do justice to all your questions. So please, some people will be asking some questions there, Dr. TG, so maybe you can find time to possibly some that you can type, but we'll definitely wait for when we have the opportunity to bring everybody together. Maybe we can now speak to them when we have the general question and answer. So thank you so much for your time once again, Dr. Yetule. So is a senior lecturer and consultant pathologist from Lagos State Teaching Hospital, and also a consultant chemical pathologist to SynLab Nigeria. So I have the honor to welcome the next speaker, which I'm going to be introducing, Dr. Tim Yin. Dr. Tim Yin is a Mindray IVD clinical specialist. So once again, he will be speaking to us about chemical analysis, better medical practice to COVID-19 treatment. So join me as I welcome Dr. Tim Yin to this great webinar. Thank you.
Good day, ladies and gentlemen. My name is Tim Yin. I work in Mandry as the clinical academic specialist for biochemistry product line. Well, it's a great honor to share this information about COVID-19 lab tests and our products. The topic, well, for today's sharing is biochemical analysis, better medical practice to COVID-19 treatment. Well, the contents are mainly containing a brief introduction of COVID-19 laboratory investigations, biochemical tests highly related to this pandemic, and COVID-19 related performance of our biochemical products. Okay, let's move on to the first section, laboratory investigations of COVID-19. Well, we know that COVID-19 is actually a viral infection, and the pathogen is SARS-CoV-2. Well, just like all the infectious diseases, there are specific tests to help doctors identify the specific infection. Well, in another word, to make the diagnosis. But for this one, COVID-19, the essential role of clinical laboratories in this pandemic extends way beyond etiological diagnosis of SARS-CoV-2 infection. The diagnosis is a starter, and the following therapeutic management, the main cause, are much more significant to some extent. Laboratory tests, which are not only target to SARS-CoV-2, to the virus, I call them ordinary tests, well, compared to specific tests, could do more. Well, for example, assess the patient's status. Well, the patient is a mild case or a severe, even critical case. To predict the patient's outcome in the beginning, and to monitor the patient's response to the treatment and help the doctors make further medical decisions. Okay, let's check the specific laboratory investigations. There are typically two kinds of specific investigations. Capture the structural components of the virus, for example, NAAT or PCR to test genome RNA of SARS-CoV-2, antigen test to identify proteins of the virus, protein S, M, or protein N. And also, there are methods to test the host immune response once an individual is infected by SARS-CoV-2. For example, the antibody tests, which target to SARS-CoV-2 specific antibodies, IgG, IgM, IgA, and so on. For a suspected COVID-19 patient, the major purpose of specific parameters is to help doctors make the diagnosis. Okay, let's move on to the ordinary laboratory investigations. The response of a human infected by SARS-CoV-2 is not only immune response, well, obviously. Cellular components could change significantly. For instance, we all know that lymphocytes decrease in COVID-19 patients, as well as platelets changes. It's a similar story to the products of human cells, no matter physiological products or pathological products. CRP, ferritin, D-dimer, LDH, electrolytes, cardiac troponin, cytokines, etc. Well, in my sharing, I'd like to focus on the top 4 parameters: CRP, ferritin, D-dimer, and LDH. Biochemical monitoring of COVID-19 patients through in vitro diagnostic testing is critical for assessing disease, the severity and progression, as well as monitoring therapeutic interventions. And we can see from this chart that, well, from the infection to the end of this entire disease process, no matter the patient's symptoms are mild or severe, and no matter the patient's in viral phase or in the host immune response phase, the 4 parameters are all clinically significant. Well, for example, we can see that ferritin, D-dimer, LDH, ferritin, D-dimer, LDH, CRP, and ferritin, LDH, D-dimer, and so on. Well, we can also see from this chart that for the relatively early stage of COVID-19, these 4 markers are quite useful because they change as the disease progression. They could be applied in assessment of patient status, monitoring of patient's therapeutic response, and predicting the patient's outcome in the beginning. Well, actually, these are exactly the clinical significance of all the ordinary parameters. Well, let's move on to CRP and ferritin. CRP, C-reactive protein, and ferritin are both known as acute phase protein. Such proteins have been shown to increase in response to stress. Well, for example, the viral infection. And both of them could increase in response to inflammation and cytokine storm. And we know that inflammation and cytokine storm are all related to COVID-19. Okay, let's see next parameter, D-dimer. D-dimer is a fibrin degradation product, a small protein fragment present in the blood after a blood clot is degraded. This net shown here is the cross-linked fibrin polymer, a unique structure in a blood clot. The small structure is D-dimers, well as shown in this picture, and it's so named because it contains two D fragments of the fibrin protein joined by a crosslink. Well, because of such features, if we could detect D-dimer in the blood circulation, we know that blood clots are formed in this patient. Well, thrombosis, to be more specific. For COVID-19 patients, especially for the severe or critical cases, increasing D-dimer is commonly seen. This parameter is a crucial marker for the management of these patients. For LDH, so far we have known that LDH is an enzyme involved in energy production, which means LDH could be found in almost all cells in the body. Tests that measure the concentration of LDH in the blood are commonly used to monitor tissue damage. Well, such kind of tissue damage are commonly associated with a wide range of diseases, and this makes LDH an important marker in COVID-19 management, because for COVID-19 patients, tissue damage, organ failure are also very common in severe cases. Cytokine storm and severe illness. I suppose we've already pretty familiar with this term, cytokine storm. It's an acute hyperinflammatory response that may be responsible for critical illness in many conditions, including viral infection, well, such as this one, SARS-CoV-2 infection. The phenomenon has been found in many COVID-19 patients. Critically ill COVID-19 patients experiencing cytokine storm, and these phenomena are believed to have a worse prognosis and increase the potential of this patient who could be die in the end. As for the four parameters, CRP and ferritin are related to cytokine storm by themselves. D-dimer is a marker of coagulopathy, and increased LDH in blood circulation means tissue damage and organ failure. They are really significant in COVID-19 management. Okay, biochemical tests. I'd like to show detailed information about the clinical significance of these parameters: CRP, ferritin, D-dimer, and LDH. For CRP in COVID-19, we can see that from the bar chart, the more severe the patient with COVID-19, the concentration of CRP is much higher. Critical group is higher than severe group. Severe group is higher than moderate group, and so on. And for a specific patient, CRP concentration increases as the disease is getting much worse. And also, for patients with COVID-19, we can find that if a patient is treated properly, the patient is respond well to the therapy, CRP concentration decreases. And for patients who are not well treated, like the control group, CRP remains in a relatively high level. For all severe cases of COVID-19 patients, the outcomes are either recovered or unfortunately dead. We can find from this study published on Lancet Rheumatology that patients who survived from COVID-19 have relatively lower CRP concentration compared to patients who couldn't survive in the end. As for the medical practitioners, when we are treating COVID-19 patients, we could know from the test results of CRP that those with constantly high CRP could have better outcome, and those with decreased CRP concentration after treatment could getting better and well, with a greater chance of recovery. Okay, this is some findings and advice from some publications. For assessment and well, the prediction of prognosis, CRP for the COVID-19 patients, it could progressively increase as the disease progression. And also for CRP who are larger than 34 microgram per liter, plus age over 60 years, indicating high probability of mortality in 12 days. As for monitoring, well, it's advised that CRP should be orbitant at baseline and repeat as indicated. Ferritin, well, it's kind of similar to CRP as both of them are acute phase proteins. Well, we can see that higher ferritin concentration means worse outcome. As reviewed by this chart, patients who died because of COVID-19 had a much higher serum ferritin concentration from the beginning of the illness to the very end. And for patients who recovered, the ferritin concentration remained in a relatively low concentration as shown here. And also, ferritin is a good parameter, a very good marker to monitoring the treatment. If a patient is respond well to the treatment, we can see from the charts, ferritin concentration decreases to a relatively low level. And this is some findings and advice from some publication about ferritin. For the assessment and prognostication, as I've already introduced, ferritin is very useful. And because of this, it's suggested that ferritin should also test in the beginning to establish the baseline and then repeat as indicated. Okay, D-dimer. D-dimer is kind of the most important parameter of COVID-19 management. Higher D-dimer concentration in blood circulation means worse outcome. Well, as reviewed by this chart, patients who recovered from COVID-19 has a very low level of D-dimer concentration. We know that D-dimer is a marker of coagulopathy, the dysfunction of blood coagulation. Well, let's see the red line. It shows the dynamic changes of patients who die because of COVID-19. D-dimer increases, and even the patient is treated, the coagulopathy is still getting worse, and the D-dimer concentration is getting, well, much more higher as the patient is getting much worse. Additionally, we can find something else from this chart. We can see triangles is the D-dimer. And also, for patients who had severe COVID-19 in the end, the concentration of D-dimer is getting higher and higher as the illness deteriorates. But on the other hand, triangles are still the D-dimer concentrations. Once the patients are properly treated, for instance, using anticoagulation therapy, well, for this case, it's using low-weight molecule heparin. D-dimer concentration decreases and the patient recover in the end. Well, for D-dimer in COVID-19 management, assessment and prognostication as I've already introduced, they are very significant. And as for monitoring, the suggestion is test in the beginning and repeat D-dimer test every day. Well, we know that anticoagulation therapy is quite important in severe and critical COVID-19 treatment. And that's why such patients are recommended to repeat D-dimer daily. Well, for LDH, higher LDH concentration means patients have relatively worse outcome, especially for these patients whose LDH are getting higher even they are admitted and treated in the hospital. As for patients recovered in the end, we can see that for one thing, the LDH concentration are lower than those who couldn't survive. For the other thing, their LDH has a decreasing trend like this one. Well, no matter for severe COVID-19 patients or non-severe patients, we can find from this chart that once these patients are well treated, LDH concentration decreases. Also, there are some findings and advice for LDH in COVID-19. Well, this parameter, LDH, is also suggested to test at baseline and repeat as indicated. Okay, let's move on to the last section, COVID-19 related performance. Well, I'm not a fan of listing all the figures and tables, so I choose two biochemical parameters, D-dimer and ferritin, as examples to highlight some features of our products. And these features happen to related to COVID-19. Well, to ensure laboratories can apply our biochemical parameters and ensure reliable test results. For D-dimer, for one thing, they are all fluid reagents ready to use. And the onboard stability is 28 days. It's quite long, and this means less reagent waste, even for a laboratory with not that many samples to test every day. As for linear range, this is also quite remarkable for our biochemical D-dimer tests. When we compare the upper limit of us to coagulation methods in FEU units, 96 microgram per microliter versus less than 20. Well, we can see that the difference is really significant. And as I've already mentioned, patients who couldn't survive from COVID-19, many test results are higher than 20. A method with a higher upper limit is much more cost efficient and well, can provide the results without without retests, and could report the results much earlier than other methods. The TAT time the turnaround time is quite short. Additionally, many COVID-19 patients are reported with hemolysis. Well, not that severe like these, but also, well, for hemolysis, it's pretty common in COVID-19 patients, especially for patients with severe COVID-19 illness. Well, and the Mandry biochemical D-dimer analysis has higher capacity of anti-hemolysis interference. All right, let's move on to ferritin. We can see that from this chart and this table, Mandry ferritin has a relatively high upper limit of linear range. This means, well, when using this test method, under many circumstances, well, they don't need to do the dilution, and there will be less rerun. And we did a survey in 4 top hospitals in China. The bar charts show the proportion of patients with ferritin concentration higher than the upper limit of the linear range when using different reagents. And we can see that, well, if the upper limit is higher enough, less samples require dilution and rerun. Well, for most of the ferritin tests, they are using immunological methods. Hook effects can occur when antigen concentration is higher than antibody and brings falsely low results. And we can see that mandatory ferritin reagents has higher hook point and a wider safety reportable range. Okay, let's do a brief summary. Well, we know that for these biochemical parameters, CRP and ferritin, they are related to cytokine storm. And this is relatively common in severe COVID-19 patients. As for D-dimer, it's a marker of coagulopathy. And this is also a common comorbidity of severe COVID-19 patients. Well, as for LDH and other ordinary parameters, they could reflect the existence of tissue damage of multiple organs. And this is also common in COVID-19. Well, as for the functions of biochemical parameters in COVID-19, they are mainly used to assess the severity of the disease, to predict the patient's outcome, and also to monitor patients' response to the treatment. Well, for all these parameters, especially these 4 I mentioned, mandatory biochemical reagents has a good performance to test COVID-19 samples. Okay, this is the entire sharing. Thank you for all your attention. Have a nice day. Thank you.
Yeah, thank you so much, Dr. Tim Yin. That was so awesome. It was a great eyes opener because a lot of things are actually coming up here which are very useful. And I've started reading a lot of comments from the participants. And I also want to encourage all our participants across the globe, wherever you are, please type in your questions. We'll be taking question and answer after the last presenter, which will be coming up with now. So thank you so much, Dr. Tim Yin, for the wonderful presentation. Dr. Tim Yin is a mandatory IVD clinical specialist. So we really appreciate you for your time, sir. So I have the honor to invite Mr. Kelechi Ekwawa. Mr. Kelechi is an IVD product specialist for Mindray and Nigeria. So we'll be taking us on the Mindray chemistry solutions introduction. So I have the honor to welcome him to this special webinar across the globe to make his presentation. Thank you and welcome, Mr. Kelechi.
Hello. Hello, good morning, good afternoon, everyone. My name is Kelechi Kenneth Ekwawa, and I am the IVD specialist, product specialist for Mindray in Nigeria. And I'll be taking us on Mindray chemistry solutions, and of course our BS430 system. But before I go on, I want to especially thank Dr. Shorion for a very great presentation, which he has actually made. which is a very good pointer to what we are going to be looking at for in this Mindray chemistry solutions. Thank you also to Dr. Tim for your wonderful presentation. So without wasting that much of our time, you are all welcome to this webinar once again. Now we'll be looking at Mindray chemistry solution for this region. Now, in the year 2004, that was actually the year where the first chemistry automated analyzer was actually first manufactured in China. And of course, yours truly, Mindray, is actually responsible for such a first China chemistry analyzer. The name of the analyzer is actually BS300, and of course it is actually well recognized, as you can see on the slide, by the Chinese regulatory. Now on this slide we can see that Mindray chemistry solution actually provide a diverse range of products that actually covers the what every institution actually needs, right, ranging from the low end to mid end and high end hospitals or labs. Now that's actually at the benchtop analyzers, as you can see on the lower left side of it. You'll find that they are usually benchtop, and of course they have a smaller footprint, so it doesn't occupy so much space. So it actually focuses on actually the hospitals that are in the primary care sectors or centers that are actually having less than 300 to 400 tests per day. Now for the second level, which is the mid-level, where we have our standalone chemistry analyzers, it actually covers for the small and medium-sized laboratories and hospitals that are actually boasting of between 400 to 1,200 tests per day. And of course, the chemistry analyzer, which will be focusing on its unique features, is actually BS430, as you can see in the central point here. Also, Mindray chemistry solution also covers for the high end. High end in this case, you are talking about the teaching hospitals, the core labs, that actually generates upwards of about 1,000 to 10,000 tests per day. So we have our modular integrated systems that actually can cater for such category of laboratories based on their throughput as well. As you can see, we have diverse range. We have BS2000, and we have South 9000 and many other ones. Now, when you consider throughput, we have different three categorization: low throughput, medium throughput, and of course, large throughput. Throughput in this case is actually a number of tests per hour. As you can see on the left side of your screen, we have about 35,000 units of benchtop analyzers that are actually catering for 100 to 300 tests per hour requirement. Of course, there are medium throughput analyzers. In this case, are standalone analyzers, of which BS430 is one of them. We have over 30,000 units installation around the world. For the large throughput, which covers for the system integrated modular systems that actually does greater than 800 tests per hour, we have about 4,500 units installed around the world. Also, Mindray Laboratory Solution actually provides a solution for medical institutions of all types that can cover entry-level labs, in this case, small labs, then mid-sized labs, and of course, for the central or those labs you will see in the teaching hospitals or the lab chains, as you can see. Now, as you can also see on this, see that this picture depicts that Mindray have broad spectrum of models that can actually satisfy the needs of all these a total lab solutions that we actually called M6000. This particular total laboratory automation line actually provides the pre and the post-analytical phases to ensure and guarantee quality and standardized resulting across all levels. Of course, many of us will agree with me that pre-analytical stage and post-analytical stages of analysis actually pose a huge major challenge in the quality of result outcome. That is why also Mindray has a total automation line like this to ensure that you standardize those steps to ensure that you have good quality result outcome. Now, what is actually responsible for what the unique technology that Mindray has been able to develop in the past 30 years of its operational existence? We have more than 200 research and development staffs in chemistry analyzer department system. About 95% of this more than 200 research staff actually holds either master's degree or they are PhD. Of course, based on their unique critical work which they do, Mindray has been able to be producing 5 to 10 new products per year. Of course, with an average of about 300 so far. Now, to be able to ensure that Mindray will sustain our growth in the near future, Mindray has always continued to devote 10% of its sales revenue towards research and development investment. And this will help us to ensure that the voice of our customers, the voice of you, the voice of those end users are actually taken into consideration in order to make sure that we can produce a unique product that will satisfy the needs and the yearnings of our end users. Now, we Mindray IVD actually have 5 independent chemistry reagent research and development labs. These labs actually help to ensure to standardize and give quality product outcome for our end users. The first among the list is actually our raw material research lab. This research lab is responsible, analyze and select the best of raw materials that is required to produce the reagents to be able to, perform top-notch result outcome by the time our end users are using them. Now, on the product development research lab, they are responsible for reagent formulation. Now, this research lab actually focuses its attention to ensure that the formulation is done in such a way that the performance of those reagents will be matching standardized guidelines meeting clinical requirements. For process development research lab is actually focused on product technology advancement, development. So this lab actually focuses on ensuring that different processes, different technologies, different patent technologies are always improved upon and optimized to ensure that a user-friendly and a better outcome for our end users, which are all of us. Also, the performance verification research laboratory actually focuses on ensuring, like Dr. Shori made mention about standardization of processes in the lab to ensure and guarantee results. So Mindray has performance verification research laboratory that ensures that every reagent that you see at your end to be used to be run tests, all of them actually passes through verification process to ensure compatibility of result outcome with the software that every model of Mindray analyzers are made of, so that you'll be able to have the best outcome. Also, reference laboratory actually focuses on ensuring that we utilize the reference material of course, utilizing the reference procedures to be able to generate, master calibration values. Of course, with these master calibration values, we'll use it to assign those values that the commercial calibrators actually have, so that you'll be able to ensure traceability to international best practice. Now, what is responsible for our advancement in our chemistry analyzer systems? These are 10 core technology platforms that are responsible to ensure that we have standardized and top-notch quality results. They are ranging from enzyme reaction technology, lattice reaction technology, lot-to-lot variation. We have an anti-corrosion technology platform. As you can see on this slide, this is actually geared towards ensuring that you can have best outcome while you are using Mindray chemistry analyzer. Also, Mindray chemistry analyzers actually adhere to strict performance verification, ensuring that our core reagent performance verification are standardized. Now, if you look at the analyzers, you will find out that we are actually more concerned also about the appearance, the reagent blanking, the sensitivity. All these characteristics will actually play a huge role in ensuring that standardization that Dr. Shorian were making references to. A particular attention, if you look at the linearity range, you find out that for every of our core reagent performance verification, linearity range plays a huge role, so that we'll know the range, the measuring range that particular that particular sample can be subjected to, without which you'll be able to get a good result outcome. Now, Mindray actually boasts of having, the more than 30,000 square meters of manufacturing sites, which actually is the biggest manufacturing reagent manufacturing plant in the whole of Asia. And of course, ensuring that we are using advanced automatic production line to ensure that we produce more than 6 million kits per year, making sure that at all times at your end user site, we will not run out of any stock of our reagents. Now, also in our manufacturing plant, Mindray utilizes intelligent reagent production line, ensuring that we have a clean room that guarantees strict adherence to pressure, temperature, humidity, dust particles, and bacteria. Of course, this ensures that the quality of the reagent is actually maintained. Of course, the water purification is also ensured to monitor the resistivity of different water purifiers. Of course, we have on the automation line, the mechanical hand can put the bottle to weighing scale smoothly. The weight testing precision can be to, of course, to the precision level of 0.01 gram, as you can see. Of course, we have a technology that also checks the bottle positioning and ensure that the cap is screwed properly to avoid the leakage of liquid. Also, we have an auto labeling system that ensures that each bottle labels are placed in exact position to ensure that we have a better outcome. Of course, we also have a system of camera that checks the barcode printing quality, ensuring that every wrong barcode printing that is done is actually swept aside, ensuring that also your Mindray analyzers recognize all reagents as you have loaded them. Also, following in the line of that intelligent reagent production, we have a real-time reporting system that ensures that we monitor the production line so that we can have optimum reagent production outputs. In this case, we have more than 1,500 sensors that actually give off real-time information Of course, those information that we are collecting, of course, we are doing that at the speed of more than 100,000 data collection, per minute. Of course, ensuring that we have more than 5,000 variables under control. So with this, you can see that this provides an insightful decision with real-time quality and execution information, guaranteeing a better balance for efficiency and quality. Now, our manufacturing plant actually boasts of complying with international best practices. As we can see here, we have obtained the ISO 9001 and ISO 13485 certificates, which guarantees that we can manufacture our products following international best practice. Now, let's focus on our BS430. Our BS430 is a unique chemistry analyzer system that that was manufactured recently, and of course has been pushed into the market in response to the voice of our customers. As we can see, it's actually a compact standalone analyzer system measuring about 1050 by 580 by 595 millimeter. Of course, it has a considerable throughput of about 420 tests per hour with an upgraded function for large capacity, onboard hemolysis capability, uniform reagent. In this uniform reagent, it means that for a center that is already have benchtop analyzers, you will be able to also use those reagents that you can use in those benchtop in this BS430 analyzer. So it means that if you have BS430 as your main analyzer, you can also have the benchtop as your backup system. You can actually interchange the use of those reagents without any problem. And now BS430 is actually for small labs that are actually to say that your throughput are already progressively going up, and you want to also match your speed of result output with action. So BS430 will be a very good product for you to consider. Of course, those centers that are having 30 to 150 samples per day, or what you can actually interpret to be 200 to 800 tests per day. Of course, centers, of course, that are doing benchtop, that are moving from benchtop to standalone system, or if your center is actually using an open system and you want to have the best result outcome, of course, you want to move to a closed system that ensures more traceability than an open system. So, ladies and gentlemen, let me introduce you to BS430 analyzer, as you can see here. Now, this is actually on the operational surface. You will see that it has a dedicated reagent probe, ensuring that, of course, the sample contamination of reagent is actually completely eliminated because it has a dedicated reagent probes. And of course, it has the two mixer heads that actually ensures the lower carryover capabilities. Then you have also a dedicated sample probe that actually ensures that samples are actually detected accurately and without contamination from reagent. We have an 8 steps preheated auto wash system that ensures that each step are actually followed and reducing the carryover capacities of contamination. In this case, you find out that for every step of testing, it actually goes through this stage of pre-wash technology to ensure no no carryovers along the way. Now, let's look at the unique features of this BS430. Of course, it has a large capacity to handle about 102 sample positions. So it means that you can run 102 samples at the same time, having a capacity to actually run 92 different parameters of tests, because it has a capacity for you to handle 92 reagents. Of course, also the minimum, it uses actually a very minimum reaction volume, which is about 100 microliter. It actually saves you reagent. So it means that it reduces, reagent consumption. And this is actually, providing you over 30% less reagent consumption than you can have in EBA and urine. It has an intelligent probe system that ensures that you have a good liquid level detection, ensuring that collision detection. It means that your probe will not, necessarily be bending or breaking, because this probe is actually equipped with capacity to be able to detect any collision. Once that collision is actually detected, it actually will stop so that it doesn't go further to break the probe. It has also an intelligent inventory monitoring system so that you're able to know the volume of your reagent, and then you can also plan. Also, this it also have a clog detection capacity. So in this case, you see that once a clog is detected, it actually will flag it for you to know. And once you know, it will be able to actually, swing into action to respond accordingly. Like I mentioned earlier on, it has a 200 tests per hour reagent package, meaning that for those of us that have the benchtop analyzers, of course, you can also interchange reagent with this standalone analyzer. Furthermore, you can also, if you look at the considerate design that we have, you have direct solid heating system that is maintenance free. So unlike other brands that actually use a water bath system to actually provide a 37 degrees centigrade environment, for Mindray BS430, we use a dry bath, a dry solid heating system that is maintenance free, and of course a reusable cuvette. The waterfall probe system actually, avoids carryovers to the level of 0.05% accuracy. Of course, the analyzer uses a reliable grating optical system, ensuring more wavelength than normal filter system that we usually have in other brands. No extra cost to replace filters, because this grating optical system actually, lasts for a long time. So it's saving extra cost for you. Now, there's something I want to also mention. If you look at this is actually a waterfall probe cleaning system. As you can see from this picture, most other brands actually has, a system of probe wash that actually uses, that encourages carryovers. But our Mindray BS430 is equipped with a waterfall system, whereby the water that actually washes this probe flows from here, goes to the probe, and actually exits from another access. And HbA1c smart sampling system that ensures, that we can have a very good result outcome for our HbA1c. Unlike you can find in other separate you make your hemolyte outside the system and then load it into the chemistry analyzer. But in this case of our system, you don't need to do that. All you are required to do is actually to centrifuge the sample, and then you have your you load your sample. Once you centrifuge it to have a very clear serum or plasma system, then you will be able to load it in the system. The probe is actually equipped to detect the level of the packed red cell that you have already created by that centrifugation. Once it detects it, it actually picks up the sample properly, and of course you will go ahead along to produce the hemolyzate in the system. So protecting the scientist who is actually working on the analyzer. This particular technology is have a good correlation with HPLC system, and of course ensures no contamination, unlike you can have in in turbidimetric method, where you have latex contamination. And of course, like I have a strong, this have a very strong anti-interference capability, avoiding, hemoglobin variant. This is actually what gives this BS430 key unique features, and we want to ensure that our end user have the best of experience with that. Also, we have what we call Mindray AAA system integration that ensures that the reagent that is actually used, the analyzer and the calibrator are all coming from Mindray. As you can also agree with me that traceability of resulting requires a closed system. And in this case, Mindray AAA system integration ensures that we can have a perfect match of the reagent in addition to the operating software that is actually matched up alongside the calibrators that are also manufactured by Mindray. On the reagent side, you can see that reagent performance verification on each model are guaranteed, and we use it to also set up and optimize reagent application parameter for each model. This actually ensures a quality result outcome. Of course, in this case, when you look at the analyzer itself, each analyzer models are actually set up in such a way, updated and verified with a database that is compatible with the reagent. Without this, you find out that you have a system whereby your result may not be correlating. But with our AAA system, the result outcome is actually better correlating. And of course, this also helps in making ease the troubleshooting system to be guaranteed. Also, on the calibrator side, you find out that the calibrator value assignment and verification for each model are actually guaranteed, and is actually traceable to a reference material, like Dr. Choria had actually mentioned earlier. Also, this actually also helps in monitoring the accuracy of Mindray systems, of course, through participation in EQAs around the world. Now, Mindray biochemistry reagents, we have about 64 different reagents that actually cover diverse, categories of clinical cases, where you have the renal, the lipid, the cardiac, diabetes, anemia, pancreatitis, as the case might be. You will see that it provides reagents on all of them. Now, I want us to look at a very powerful software system that is equipped in the BS430. It has an intelligent operational system that ensures that you can have a auto rerun, meaning that if you have a system, a situation where that requires that you rerun immediately, the analyzer is equipped with capability to ensure that the auto rerun is actually run properly without any issue, meaning that when you now have an abnormal sample or abnormal result outcome, the system is actually equipped in such a way that you can auto rerun to recheck and double check and be sure that the result will be good for you to actually release. And of course, it's equipped with one key parameter import system. This means that with one button press of a key, you can import all the parameters at one go. You don't need to be configuring it manually by yourself. Of course, it has a stepwise maintenance guide for you to actually conduct your daily, weekly, monthly maintenance requirements. Of course, it has also capacity for twin tests, meaning that you will use one reagent pipetting capacity for you to actually run two different tests. For instance, in the case of hemoglobin and A1C assessment, you will find out that you are not required to do double aspiration of reagent, but in this case, you will just do single aspiration for you to produce two such results. It has also a reflex function that actually ensures that for abnormal cases that you are actually informed for you to ensure that a good action is taken to make sure that the result is good before it's actually taken out. Then it has an auto QC management system that ensures that your QC can be preset or can be pre-programmed for you to actually run without missing out anyone per day. It has also a serum index system, a reagent test management system that ensures and monitors your inventory system. Now, on the basic level of the AAA system, it actually encourages, it is actually founded based on closed system algorithm that ensures that the basic and advanced to the operational software so that you will be able to get a very good result outcome, as you can also see on this. This ability helps to solve clinical abnormal samples. Now, also, as you can see on this slide, with BS430 is actually having an interface that is user-friendly, and it has a very short startup time. As you can, if we compare it with other brands, the time to system restore or the time to system initiation, in case if you're having a challenge along the way, you find that this is actually a very faster time, more than other brands, as you can see on this slide. Now, this is actually Mindray traceability chain. At the top of the chain, you will have, reference method where that involves the reference material and of course, following the reference procedures. These reference procedures are followed to be able to generate what we call Mindray master calibrator. These master calibrators actually are used to, pass through standardized processes to ensure that the commercial calibrators that you eventually get at your site actually is assigned values. Those values that are assigned to those calibrators are very closely traceable to the reference material. So you can be very well assured that the result traceability is also guaranteed. Now, Mindray also, has measurement capability of the reference lab has recognized the world over. Our reference labs, like I have mentioned, were qualified to provide the value assignment services. Of course, as you can see here, in this reference lab was actually established in 2007. It was actually a first reference lab in Chinese IVD. And then, of course, Charlie has participated in a lot of, international authorization systems, as you can also see on the left side. Now, these are actually the traceability documentation required regarding our parameters. As you can see, the establishment of standardization is actually in line with international reference centers, as you can see, has been recognized for Mindray. Also, this is actually a certificate of traceability to ensure that our HbA1c is actually directly traceable to DCCT reference and IFCC reference as well. Now, this is actually some of the EQ performances of our analyzers that are scattered around the world. With the technology of our AAA matching system, we ensure that better performance of resulting is actually done for your EQA performance. So it means that you can actually get a better outcome following our AAA matching system. Also, modular systems, which we call serum automation modular system. In this case, we have SAL 6000. SAL 6000 is actually a combination of biochemistry, BS4800, and of course our CL2000I systems, plus our sample processing line 1000. This sample processing line is actually what guarantees automated sample loading system to ensure that the system is actually fully supplied with samples per time. Also, we have SAL 9000, which is a combination of our BS2000 and CL6000I, of course, equipped with and supported by our sample processing line 2000. So this SPL2000 means that it can actually handle about 2000 samples per time, while SPL1000 can also handle processing of 1000 samples per time in terms of feeding to the analyzers. Of course, with this modular integrated system, it means that for every patient, we are going to use one tube to actually run the testing for that patient. And of course, it can be by one operator, and of course, through one system generating one report. And of course, that report actually is robust, is traceable, is accurate, and of course, intelligent. Now, these are some of our Mindray reference sites for our serum automation lines. As you can see, we have them in Italy, Argentina, Thailand, India, Turkey, Chile, Romania, Morocco, Saudi Arabia, and many other places, as you can see. We are hoping that very soon, one of us listening to me today will be interested to ensure that we can have a SAL series in Nigerian market. Now, these are also some of our Mindray reference sites where you, for our SAL series, Kolobuti Hospital in Ghana has it, Al Samad Group in Oman, Laboratory Alta in Morocco, of course, King Abdullah University Hospital in Jordan, of course, in Sudan, and of course, in Angola also. They all have our SAL system. I want to thank you very much for listening to me, and I will be welcoming your questions where necessary. Thank you.
I want to really appreciate the people that have come together to put this together, the West Africa Postgraduate College of Medical Laboratory Science, the Association of Medical Laboratory Scientists of Nigeria, the Crown Healthcare, and Mindray for bringing this very, very educative webinar together for all the participants across the globe. We really want to appreciate all of us that have participated from every part. I've seen a lot of my friends, colleagues. I've also seen new people here. So we really appreciate you for your time, and we'll let you know when the next webinar will be coming forth, and we'll keep in touch with you. And for certificates, of course, there will be certificates. Just ensure that I hope you're all registered when you are logging in. So you are going to be, we have a system that can provide online certificates for what you've attended. So we really want to appreciate you. We want to appreciate all the team, all the speakers, from the Dr. GCO Okara to Dr. Yotungi, the first presenter, Dr. Tim Yin, Mr. Kileshi Ekowa, and my fellow colleagues, Jason, that have been make sure working from the background to see that this become a success. You will not believe that it actually happened. So we really want to appreciate you so much. All the participants, you are the king. We appreciate you for taking your time to attend this webinar. So see you soon on the next line of the webinar that will be coming to your table. It's going to be very, very much interesting. Just watch out as we bring this to your doorstep. So thank you so much for your time. For now, I say goodbye and see you again. Thank you.
Thank you all. Bye.
Thank you.
Thank you.
Thank you.